Hello everyone, my name is Charlotte, and this year I studied a striped mullet die-off event that occurred in Malibu Lagoon in 2018. Now, a bit of background on this event. It occurred on August 27th in 2018. Over 6,000 deceased striped mullet were found, and another die-off occurred 3 weeks prior in Ormond Beach Lagoon. And here is Ormond Beach Lagoon versus Malibu Lagoon. Now, the striped mullet is a fairly regular fish to see within lagoons or within the ocean. They are around 9 to 20 inches when they're an adult, and they're seen as a very hardy fish because they can live in very large temperature, salinity, and dissolved oxygen ranges. Malibu Lagoon, where the main die-off occurred, was restored twice, once in 1983 and once in 2013. Although the restoration is not the only thing impacting lagoons, continued urbanization around the lagoon has continued to constrain this lagoon. If we can see in this image, there are houses on either side. There's the PCH bridge that goes over Malibu Creek, and there are businesses continuing up Malibu Creek, continuing to constrain the lagoon. When we begin to look at the die-off, here is one individual from the Malibu Lagoon die-off, an individual from the Ormond Lagoon die-off, and an individual from another die-off, the Oso Flaco Lagoon, which was in 2017. So these were all recent die-offs that occurred in Southern California for the striped mullet. Once I began researching this, I realized that all three of these individuals had interesting red tinging along the fins and the gills and other exterior parts of the body. I began researching into what could have caused this, and hemorrhaging was the main cause. So hemorrhaging, there's a lot of factors that can cause hemorrhaging. One of the main is hypoxia, which is when the body does not get enough oxygen. There are many ways that this could occur. One is from a heat wave, which did occur right before the Malibu Lagoon die-off and during the Ormond Lagoon die-off, which would cause increasing water temperature and therefore decreasing dissolved oxygen content. An overnight low dissolved oxygen could occur through an increase in transpiration of algal species within the lagoon. A harmful algal bloom could occur where eutrophication occurs, decreasing the dissolved oxygen in the water. And other factors can cause hemorrhaging, such as contamination or a disease or pathogen within the water. To start testing how hypoxia could have occurred within the lagoon, I looked into different atmospheric conditions, such as temperature, wind, and precipitation. And I compared the conditions in 2018 to different years through 2005 to 2020. I also looked into the Malibu Lagoon conditions, such as water temperature, dissolved oxygen, pH, and oxidation reduction potential. I got all of this data from either NOAA or the Malibu Lagoon Restoration Enhancement Project, where they were testing to see how the lagoon reacted from the restoration that occurred. If we begin to look at the temperature comparison within Malibu Lagoon, here's the first graph. It is temperature maximum on the y-axis, date on the x-axis. We see here in black is 2018, and then behind in gray is all the other years. And then in this red line specifies the Malibu Lagoon die-off, which again was August 27th. So here we can see that in early July, there was a very large spike in temperature. And after that, it was a continued high temperature. I enlarged that portion of the graph to make it a little bit easier to read. So here is early August, the same temperature maximum on the y-axis. We can see that it starts high, and then by August 12th or 13th, it began to decline. And then the Malibu Lagoon die-off occurred. We can see the same thing here for the temperature minimum. In early July, it is high and it stays continuously high until August 13th. Now, next we can look at how this atmospheric condition impacted the water within the lagoon. So within the final comprehensive report, there were a lot of different stations impacted. I chose station 8 and 2 to look at today because they show different viewpoints of what occurred in the die-off. So here we have station 8 and station 2. Again, water temperature and dissolved oxygen are on the y-axis and date is on the x-axis. And this black line is the Malibu Lagoon die-off. So here we can see for water temperature first, in both graphs, we can see it's around 27 degrees Celsius, and then it declines a bit to 25 when the die-off occurs. So the temperature limit of the striped mullet is around 20 to 32 degrees. So this shows that during the die-off, the temperature limit was not above what the striped mullet can have, but considering the heat wave began in early July, this was a very prolonged period of time for these fish to have a high temperature. So this would have caused a lot of stress on the fish if it was above the temperature limit or not. Next, if we look at dissolved oxygen in red, we can see in this graph, interestingly enough, it actually increases. And then at station 2, it is fairly low. So with this, we can see that in Malibu Lagoon, there can be very differing dissolved oxygen points depending on where we look. But there were low dissolved oxygen points in the lagoon. Now we can move back to atmospheric conditions. Here we can see wind versus date. And as we can see, no crazy large wind events occurred. If we look at precipitation, literally no precipitation events occurred, and this seems regular since there's barely any precipitation events in 2005 through 2020. So we know since there were no quick changes with wind or precipitation, they would not have altered the lagoon conditions. Lastly, we'll move back to water conditions within Malibu Lagoon. Today, we're not going to be looking at the ORP, we're just going to look at pH, which is in green. Interestingly enough, here we can see after the die-off, there was a quick increase in pH in both of these. And it's interesting because this could have been a consequence of the die-off, or it could have been a factor within the die-off. And we'll know more about what geochemical processes occurred to create this pH jump when we start testing eDNA. So eDNA is actually a very big part of the research that I'm doing right now. Unfortunately, with COVID, we are unable to get the results by the end of the year, but hopefully in the summer or next fall, I'll be able to get these results and I can look at specific diseases, algae, and bacteria that were in the lagoon at the time and look at other population ships other than striped mullet that changed during the die-off event. And lastly, I want to continue to relate the die-off to large events such as climate change, restoration, and urbanization, and understand how these stressors impacted the lagoon and the species within the lagoon. To end, I just want to thank Rachel Turba and Dr. Jacobs for being great project mentors. I want to thank the Sustainable LA Grand Challenge Undergraduate Research Scholars Program, and most notably Dr. Rebecca Scheip for all her help this year, and the Cal eDNA internship program for helping me start doing research this last summer. So thank you all so much.